Ladies and gentlemen, Dr. Alice Shaw.
Yes, yes.
I want to just start off with this slide, which I often show in my talks to other oncologists, really illustrating how amazingly quickly this field of ALK-positive lung cancer has moved. And I know many of you have experienced this actually in real time. I've experienced it in real time. And so it is pretty incredible to think about the ALK inhibitors that we now have available to us for patients who have ALK-positive lung cancer. With the first one, of course, the first generation ALK inhibitor, crizotinib, being approved back in 2011. So think about that. That's actually exactly 8 years ago this month that crizotinib was approved. And then, of course, we all know that there soon came second generation, more potent ALK inhibitors, first developed in the setting of crizotinib resistance. Those were all approved, as you can see, ceritinib, alectinib, and brigatinib. And then most recently, of course, we now have a third generation, even more potent ALK inhibitor, lorlatinib, newly approved for patients who have been on prior ALK inhibitors. So it is pretty incredible to see 5 ALK inhibitors in the span of just about 8 years, not just developed and studied and tested in patients, but actually now fully approved. And many of these drugs are actually widely available, not just in the US, but outside the US for ALK-positive patients. So for today, I wanted to, there's a lot that's happened, and I kind of had taken a poll of my ALK-positive patients in my clinic and asked them what they wanted to hear about, because obviously I can't talk about everything. I could actually stand here for 8 hours or 10 hours talking about ALK, but obviously I only have about 30 minutes. So I'm going to focus on a couple areas, and then of course we have a Q&A, so we'll have plenty of time to actually address some of your specific questions if I don't get to them. So I thought I'd start off with a really quick intro to ALK. I know that some of you are really ALK aficionados, but others are very new to the ALK field, so I just want to do a little background on ALK, and in particular just highlight the drugs, so everyone's familiar with the drugs that we'll be talking about. And then really I want to address 3, I would say, sort of hot topics, at least amongst patients I've spoken to recently. And those 3 topics have to do with the importance of repeat biopsies. When do we do them? Why do we do them? Do we do tumor biopsies? Do we do liquid biopsies? I want to also talk about the ALK Master Protocol. Many of you have heard of this, and it's a recently launched protocol, and there's not that much information out there yet, so I wanted to share why an ALK Master Protocol is being done, what we hope to learn from it. And then the third topic I want to address has to do with immunotherapy, again, specifically for ALK, because all of us have seen those ads for Keytruda and Opdivo, and we've all wondered about why does it keep saying, not for ALK-positive patients. So we'll talk about immune therapy, checkpoint inhibitors in particular, and actually how I think we should really move the field forward in terms of the immune system in ALK patients. And then finally, I am going to end on what we're working on now and what's coming up for the future of ALK in terms of research strategies and new approaches. Okay, sounds good? Okay, so let's start on the background, and then we'll move to the hot topics. All right, so many of you may remember that ALK was discovered as an oncogene in lung cancer specifically, actually 12 years ago this month, and it was discovered, this rearrangement involving ALK was discovered by Dr. Hiro Mano in Japan, and he identified this novel fusion protein that includes a portion of the ALK gene fused to a partner protein called EML4, and that's only found in the cancer cells. It's not something that's found in what we call the germline or something that would have been hereditary. It's found just in the cancer cells in the lung. And this was a really incredible discovery at the time. Again, taking people back in time to when we were seeing patients in the clinic in 2007, really the only target we knew about was EGFR, epidermal growth factor receptor, and we had some first-generation EGFR inhibitors, but otherwise, if you didn't have EGFR, really we had no other treatment options besides chemotherapy. So in 2007, when this was reported, this was met with a huge amount of enthusiasm that there could be a new target in some patients with lung cancer.
This is just reminding everyone that it's not a mutation. Sometimes you hear people talk about ALK mutation. I have an ALK mutation. It's really the rearrangement or a shuffling of the chromosomes, which is the key event that activates ALK abnormally and leads it to drive the abnormal growth and survival of cancer cells. Now, soon after ALK was discovered in 2007, we were really fortunate, and this is what I often refer to as serendipity, because at literally the exact same time that ALK was discovered by Dr. Immuno as a potential target in lung cancer, we already had crizotinib in the clinic. It actually had already gone through phase 1 testing, actually even starting a full year before ALK was discovered, because crizotinib, as some of you know, was initially developed to target a different protein called MET. And it just happened to also inhibit ALK. And so when we heard about that discovery by Dr. Immuno's group, we very quickly pivoted and repurposed crizotinib as a new ALK inhibitor. And these are the types of responses we started seeing very, very early on. This is one of our early patients who has ALK positive lung cancer. You can see how much disease he had at the time in his lungs. And he had already been through like 5 or 6 lines of therapy, and then ALK was discovered. We did his testing, found out he had ALK rearrangement. He started on crizotinib, and you can see this amazing response. And I think all of you here are probably kind of used to seeing responses like this. We talk about them a lot, but back then, we really never saw responses like this outside of EGFR patients who were on an EGFR-targeted therapy. So this was incredibly exciting to really realize that ALK was indeed a real target for lung cancer, and that we already had a drug in the clinic that was showing activity like this. So that, of course, led to lots of studies. I'm not going to go through any of these studies in detail, but lots of studies looking at crizotinib, the first ALK inhibitor for patients with ALK-positive lung cancer. We had many phase 1, 2, phase 3 studies, all establishing that crizotinib was very, very active for patients with ALK-positive lung cancer. But what you can see here in that red box is PFS, which stands for progression-free survival, gives you a rough sense of how durable responses are to crizotinib, and you can see across these studies that it's in about the 8 to 11-month range. Again, that's huge compared to what we might see with chemotherapy, but still clearly not good enough. And what we started studying very early on as we started testing crizotinib is this problem that many of our patients are facing, which is the problem of drug resistance. So this is a pretty typical example of what we might see, where you have a patient who has a cancer in the lung here on the left, beautiful response, as we're used to seeing with crizotinib, and then, of course, at some point in the future, for this patient, it was about 3 years, you can see that all of these originally sensitive tumors had come back. And so that's a problem that we call acquired drug resistance. And this is when we were starting now to study what are the mechanisms behind the development of resistance. Very soon after starting to study, we and other groups had found now that there were mutations that could occur within ALK that could now hinder the ability of crizotinib to inhibit ALK. And in some patients, these mutations in what we call the sort of active site or the active site of this enzyme are shown here by the yellow balls, were actually found in some patients and could completely block the ability of crizotinib and hence now allow the cancers to grow back. And so just with a little bit of information like that shown here, many groups and companies, pharmaceutical companies in particular, really set out to develop better ALK inhibitors and ones that could overcome these common crizotinib resistance mutations. So I mentioned crizotinib as the first generation ALK inhibitor. We've talked about second generation, more potent ALK inhibitors, and those really were developed to try and overcome the most common crizotinib resistance mutations, like what we call the gatekeeper. And then even more recently, third generation drugs that are again rationally designed really to overcome as many of those ALK mutations as possible and also to penetrate into the brain to high levels. So let's just quickly walk through how we use these drugs in the clinic. So of course we talked about crizotinib already. I mentioned that the second generation ALK inhibitors, ceritinib, alectinib, brigatinib, were all initially studied in the post-crizotinib setting, received their initial approvals, and so this was a pretty common sequence. Many of our patients, in fact many of you may even have been on this sequence of being on crizotinib first, then followed by another more potent second generation inhibitor. But as we were seeing these great responses to second-generation inhibitors in patients, we were, of course, wondering, well, why not use these second-generation inhibitors first? Why are we starting with a drug that's not as potent and then moving to a more potent one, when in fact we could just start with the more potent one to begin with? So this led to a number of large studies comparing second-generation inhibitors to crizotinib. And many of you are familiar with alectinib. It's also called Alecensa. Second-generation drug, more potent than crizotinib, very brain penetrable, great activity in patients who've already been on crizotinib. And what we had wanted to study was how active it was as first-line therapy. So this is the ALK study that many of you have heard of, where we studied alectinib head-to-head against crizotinib as first-line therapy. And this was the primary analysis I had presented about 2 years ago, showing that alectinib really was much, much better. It pretty much kicked crizotinib's butt, as you can tell here, right? Greater than 50% reduction in the risk of progression. So very, very significant improvement. And this really led now to alectinib and other second-generation inhibitors really becoming the preferred first-line therapy for ALK-positive patients. So again, a complicated table, but this is basically summarizing all the data we have now for alectinib and also some data for brigatinib as first-line therapy. And again, I'm just highlighting here in that box the efficacy, and here you can see with first-line alectinib, now our median progression-free survival is pushing out close to 3 years. And remember, contrast that to crizotinib, where I told you it was more like 10 or 11 months. So a big difference when we use our more potent ALK inhibitor first. And then shown below there is what we call the intracranial or the brain response rate, which also very high for these two drugs, alectinib and brigatinib. So these have become really, I would say, kind of the definitely the preferred options for first-line therapy for ALK-positive patients. So then this brings us to the next obvious question as well. But now we have really good ALK inhibitors, even first-line, these second-generation drugs. What do we do after second-generation drugs stop working? And this, of course, really motivated us to develop these third-generation drugs that we really were developed by the medicinal chemists. This particular one was developed by the medicinal chemists at Pfizer to address all of the known resistance mutations that we had identified in patients' biopsy specimens. And lorlatinib was also developed to be really brain penetrable. So again, complicated table where I'm just really mostly highlighting that lorlatinib is really active and it covers all of these known resistance mutations. And so this then led to a phase 1 study, which we started now 5.5 years ago. This is just, again, complicated slide, but it's what we call the Waterfall Plot, summarizing tumor shrinkage in individual patients who receive lorlatinib. And then the number above each bar indicates the number of prior ALK inhibitors the patient may have received. And so you can see here in this waterfall plot that many of the patients had received 2 or 3 prior ALK inhibitors before going on to lorlatinib. Then they went on to lorlatinib and had these responses, as you can see here, by the negative or downward bars. So very active drug, even in patients who've been on prior ALK inhibitors. And then what we were very, very happy to see is that this drug, as we predicted, was also really active in the brain. This, again, is a waterfall plot on the right-hand side, now summarizing how much tumor shrinkage there was in brain metastases. And you can see, again, some patients where we saw complete resolution of brain metastases, even though they had been on prior alectinib or prior ceritinib, for example. So this really now has established what we kind of consider sort of a standard sequential therapy for patients with ALK-positive lung cancer, where they go through first, second, third generation, or more patients these days are starting on second generation and still then have a third generation option with lorlatinib. Okay. So now that's kind of the background, kind of gives you a sense of the sort of the lay of the land in terms of how we use these ALK inhibitors. And so now I want to dive into these questions. And the first question really has to do with repeat biopsies. So as I mentioned early on, even with crizotinib, we were noticing this problem of drug resistance. And really our hope was to try and understand how resistance develops, what's driving resistance. And I mentioned to you how we had discovered resistance mutations that occur within ALK itself. And the way a lot of these studies have gone for, again, our group and many others around the world is really by studying the cancers themselves. So doing a biopsy of a resistant cancer in a patient, extracting the DNA from that and now sequencing the DNA and trying to understand if there are new changes that the cancer has acquired that may account for resistance. So we've done a lot of that rebiopsying of patients, as you can see here, and doing the genotyping. But for us, where we do a lot of basic research as well, we've also used these biopsy samples to generate new models of ALK-positive lung cancer in the lab. So cell line models, as well as mouse models, what we sometimes call PDX models. And these have really been, I would say, vital for us in terms of, again, understanding resistance mechanisms, discovering new drivers of resistance, and also doing preclinical testing of new therapies to see if we can overcome resistance. So I think this model that I often show here in my scientific talks is really to emphasize how important studying the cancer itself is. And of course, how important many of our patients have been in contributing these biopsy samples so that we can understand resistance better and develop better strategies. So what we've learned about resistance, I'm going to focus primarily on second-generation inhibitors, but I'll talk a little bit about crizotinib, since again, that was our first-generation drug. And what we learned early on is that for crizotinib resistance, a minority of patients, maybe about 20% or so, have resistance mutations within ALK. Some of them are listed down here. And again, I mentioned before, these occur in the active site of the enzyme. They block binding of crizotinib. But for the most part, most crizotinib resistance is not due to an ALK mutation. The story changes quite a bit, though, when you start looking at these more potent second-generation drugs. And again, these are complicated-looking pie charts, but what I'm trying to highlight here is that when patients fail on a second-generation drug and we rebiopsy a resistant tumor, in probably a little over a half of patients, indicated by the different rainbow colors here, we do identify a resistance mutation within ALK. So it's pretty common to identify in patients who have failed a second-generation drug. And what I think you can see is that for each of these drugs, the spectrum or the pattern of ALK resistance mutations does differ slightly depending on which second-generation ALK inhibitor the patient was on. However, there is a commonality here that you can see in the red, which is that probably about a good quarter of patients, when they have failed on a second-generation inhibitor, will develop this very refractory mutation within ALK, which we call G1202R. So that was a very important one for us to focus on because it is the most common one, and you can see that it applies across the board to all of the different ALK inhibitors, but particularly to the second-generation inhibitor class. And that's where lorlatinib came in. I mentioned lorlatinib was specifically designed to overcome ALK resistance mutations. And so we were studying, I showed you the data already for the lorlatinib trial, but our hypothesis in all of this is that lorlatinib would be able to overcome resistance in about half of these patients, the ones where their tumors have developed an ALK mutation for which lorlatinib is active. And now you're probably thinking, well, what about all the other patients who don't have an ALK mutation? Because about half of patients, when they become resistant, will not have an ALK mutation. And all of our preclinical studies suggest that those cancers have become even more complicated and likely have other pathways or other proteins besides ALK activated. So simply using another more potent drug like lorlatinib is likely not going to work as well. So now to prove this, we looked at our data from the lorlatinib study. And again, complicated pictures here, but what I'm trying to highlight is that if a patient fails on a second-generation ALK inhibitor because of a mutation, then more likely than not, they will respond to lorlatinib. And the difference is shown down here. There's about a 70% chance of having a great response to lorlatinib if you have a mutation, but only about a 30% chance if you don't have a mutation. So here it's very clear that biopsies are helpful because we can now identify patients who have an ALK resistance mutation, and then we have a sense that those patients are the ones more likely to respond well to lorlatinib. And that holds up in terms of the durability of response as well. So this is looking at progression-free survival, and again highlighting that if you have an ALK mutation after a second-generation inhibitor, you're likely to respond to lorlatinib, and those responses are likely to be durable. Here, the median progression-free survival came in at about 11 months, and if you did not have a mutation, it was down as low as 4 months. So again, that mutation status from a repeat biopsy really gives us some sense, what we sometimes consider stratification, those patients more or less likely to respond to another ALK inhibitor. So then how can we use this information, especially in a patient who can't, for various reasons, undergo a tumor biopsy? Well, fortunately, many of you are now familiar with liquid biopsies, and I think that came up earlier today as well. Liquid biopsies refer to biopsies of the blood. So now looking in the blood for circulating tumor DNA. So that means DNA from the tumor itself that's shed into the bloodstream and now captured with very sensitive techniques and sequenced to look for actually both the presence of the ALK rearrangement, but more importantly in the setting of resistance, the presence of an ALK mutation. So this is just a nice, I would say, drawing illustrating how cancer cells can shed their DNA into the blood. And the reason this is so complicated is because lots of normal cells also shed their DNA in the blood. So it's almost like looking for a needle in a haystack, where you're looking specifically for the tumor DNA that's been shed into the bloodstream. But we've been able to do that. The technologies have improved incredibly the last few years. And so now we fairly routinely will use liquid biopsies, so ctDNA biopsies in patients, specifically looking for ALK mutations. So here's some work that we recently published, actually just a few days ago, in Clinical Cancer Research, where we had over 100 patients in my clinic, all with ALK positive lung cancer and all on one of these first, second, or third generation ALK inhibitors. And you can see many of the patients had been actually on 2, 3, or even 4 different ALK inhibitors. And we were starting to see resistance develop. So we sent off a blood sample for liquid biopsy. And shown here, what I'm highlighting for each patient, shown by a column, is the presence of ALK resistance mutations that were picked up by this liquid biopsy. So the one we used actually was the commercial one from Guardant. But this is also similar to some of the other ones that are available. Many of you have submitted blood samples for spacewalk. That's another type of liquid biopsy that's probably more sensitive for detecting the rearrangements. But Guardant has been very good at actually detecting ALK mutations. And so for all of these patients here, these boxes illustrate the different ALK mutations that we identified in the bloodstream and that likely were contributing to the patient's relapse on the ALK inhibitor. Here are a specific cohort of patients who have been on lorlatinib. All of these patients had been on probably at least 3, maybe 4 ALK inhibitors. And again, looking in the bloodstream for the presence of ALK mutations shown here. And what we were seeing is that as patients become increasingly resistant to second and now third generation ALK inhibitors, these cancers become more and more complex. And now you actually have multiple different ALK mutations developing, not just 1 or 2, but often 2, 3, 4, or more ALK mutations developing. And it's really a reflection of the sort of potent selective pressure that these drugs place on the cancer. So what do we do with this? I mean, one thing we can do with liquid biopsy is we can identify a mutation, and that will sometimes allow us now to pick an ALK inhibitor that we think is going to be most effective, and I'm going to talk about that in a second in the context of the ALK Master Protocol. But also what we can do is we can track these mutations over time in patients as they go from one ALK inhibitor to another. So I'm going to show you one example of one of our ALK patients who started on alectinib, and we drew actually liquid biopsies about every 3 to 4 months on this patient as she went from alectinib to lorlatinib. And I'm going to show you what the liquid biopsies reveal to us. So when this patient started to relapse initially on alectinib, we had gotten a liquid biopsy here. And what I'm showing here, you can't appreciate the whole picture yet, is the circulating tumor DNA burden. And what we identified in the circulating tumor DNA was a resistance mutation called V1180L. That's shown by the yellow. And this patient then transitioned actually at this point to brigatinib because we actually had data that brigatinib can overcome this very specific alectinib resistance mutation. So we had her treated with brigatinib, and she did pretty well. And what we could see in the blood, again, we collected a blood sample here while she's on brigatinib, and you can see this V1180 clone that caused resistance to alectinib is now diminishing because brigatinib is very active against this mutation. But at the same time, we could actually start to now detect a new ALK mutation arising on the brigatinib, and that's the G1202R, which I mentioned to you is very resistant to second-generation drugs. And ultimately, when she relapsed clinically, we could see growth of her cancer on scans. Now we could clearly see that this G1202R clone was likely driving her resistance. And so at that point, we know that lorlatinib can overcome this mutation. So we switched her to lorlatinib, And indeed, you can see she had this really nice response. We checked her midway on lorlatinib, and she had almost no detectable circulating tumor DNA. So lorlatinib almost cleared all of her circulating tumor DNA. And consistent with that, her cancer also got much, much better. But unfortunately, with some more time, you can see that she started to develop resistance. And what we could see happen in the blood is that G1202R started to emerge again. And that was in combination with yet another ALK mutation, this 1156Y. And this particular combination of ALK mutations in one cancer cell is what drove her relapse on lorlatinib. So I think this is a nice example of how we can use liquid biopsies in real time. It can be really informative in guiding the selection of targeted therapies, and it can also be really informative in discovering new resistance mechanisms, which ultimately, as we develop more and more therapies, hopefully we'll be able to match this particular patient to yet another therapy for her particular set of mutations. So let's go to ALK Master Protocol, because this is really the focus of the ALK Master Protocol is how can we best sequence our ALK inhibitors? How can we use biopsies to guide the selection of ALK inhibitors? So really this is, I would say this is kind of like a precision medicine trials, where we're actually trying to tailor therapies in the setting of resistance. So not just do your initial genotyping, look for ALK, look for EGFR. This is actually now at the time of resistance to a second generation inhibitor, how can we best tailor therapies in that setting? This is a very large study. It's sponsored by the National Cancer Institute. It's being run by one of the cooperative groups, NRG. I'm a study chair along with Shakun Malik from NCI and CTEP, and you can see we have 4 other study co-chairs, including Christine Lovely, who's here today. So lots of us who are very invested in this question of how can we best sequence our ALK inhibitors. And really, this is the bottom line of this. So if you have a patient, if you are a patient on a second generation ALK inhibitor, ceritinib, alectinib, or brigatinib, and you start to develop resistance, what is the best strategy at that time? Should we just empirically pick another ALK inhibitor, kind of just guess on another ALK inhibitor and just try that one? And many of you would say, why would we ever do that? But I have to say that in the community, when I hear from docs, and when I hear from docs actually around the world, I would say especially outside the US, they really feel strongly about just picking another ALK inhibitor, not selecting one, but actually just trying one to see if it works. And I think a lot of that's for convenience. You know, you don't have to do a biopsy. You don't have to really think about it. You just prescribe whatever other ALK inhibitor you have access to. But I think all of our studies, all of the biology, all of our clinical experience really suggests that we have to do better than just guessing. And the way to do that is by doing the biopsies that I talked about, looking for these ALK mutations, and then matching patients to the right drug based on their genetic profile. So now, why is that important? Well, here's some real examples of why I think this is important is if we have patients who have been on alectinib and now they have failed on alectinib, and we just put them on ceritinib or brigatinib. We don't do any profiling, we just put them on one of those drugs. You can see in this slide that the response, the efficacy is not so good. We're down at probably 20% or so in terms of response rates. So it really doesn't make sense to just pick another second generation inhibitor after alectinib without thinking about what's driving the resistance. Now this is what the schema looks like for the protocol, and I just want to go through it a little bit to highlight a couple key points. So it's obviously for ALK positive patients. All patients have to have received a second generation inhibitor. It could have been alectinib, ceritinib, brigatinib, or ensartinib. You could have received crizotinib before that, but you can't have gotten multiple second generation drugs or second generation and third generation drugs. We have to have some ability to kind of narrow in on a very specific question, which is what to do after failure of a second generation drug. And you can see we're aiming for about 600 patients altogether, and all patients will need to undergo a tumor biopsy and a liquid biopsy. And this is really important because we actually are trying to establish whether or not liquid biopsies can fully replace tumor biopsies or not. So it will allow us to do concordance between tumor and liquid biopsies. And then this is actually a simplified schema here, but what you can see is based on the genetic profile, based on what we know is driving resistance, patients will then be matched to drugs that have demonstrated some activity. We're not going to pick drugs where we've seen no activity. We're picking drugs that make sense based on the chemical structures and all of the activity we know about. And I would point out these are the different ALK mutations. There are more than just shown here. You may also notice here, sorry, this shouldn't be ALK, but this should just be MET amplification. For MET amplified patients, that means MET has now been, there are too many copies of MET that have been turned on, and that's driving resistance. And there's some early data suggesting that if we target both ALK and MET, that may be very effective, and we may be able to do that in the form of crizotinib because it inhibits both MET and ALK. So that's the sort of rationale for this particular pairing. And then this is a really important arm as well. For patients who do not have an ALK mutation, I mentioned earlier that we believe that those cancers are not driven by ALK anymore. They're likely driven by some other pathway that's been turned on. It could be another protein, another kinase, something else besides ALK. We think there's some activity in some of those patients to another ALK inhibitor, but in fact, those patients may do better with chemotherapy. But we don't have any data really proving that. And so this will be the first chance we have to really understand whether or not chemotherapy may in fact be preferred in some instances, and particularly for patients who have developed these more complicated ALK-independent tumors. Our primary endpoint in the study will be to look at response rates. We'll have a number of other secondary endpoints. And then again, I just want to highlight how important sort of the correlative studies in this study are, which is really to establish if we can fully use liquid biopsies. You know, are liquid biopsies sufficient and I would say accurate enough that we can be making all of these treatment decisions based on them. So this is a really important study to show that. All right, and the last topic I want to focus on is the immune system and checkpoint inhibitors. And all of you have heard about these. Everyone's excited about these across all cancer types, not just lung cancer, but pretty much every possible cancer. We've studied checkpoint inhibitors, and I think many of you are familiar with the idea behind checkpoint inhibitors, where we do know that in some cancers, the cancer is actively suppressing the immune system, specifically the T cells that could kill the cancer cells, and they're doing it through signaling this interaction here between PD-1, a receptor, and PD-L1, a ligand here. This interaction is the tumor cell is actively keeping this T cell away, and now these new drugs you hear about like Keytruda and Opdivo interrupt this signaling. So now there's not this signaling going on and the T cells can be activated. So we're trying to, in this setting, harness the immune system, unleash the immune system to fight the cancer. This has been very effective in many patients across the board, except that it's confusing still in the literature about how do we identify patients who are going to respond to checkpoint inhibitors. What we do in the field of non-small cell lung cancer is we do have a marker called PD-L1, which many of you may have heard about. Your cancers may have been screened for PD-L1. And the confusing thing about PD-L1 that I wanted to emphasize here is that in the setting of ALK positive lung cancer, PD-L1 testing is not so helpful. In fact, many of our ALK patients have high level PD-L1 and yet ultimately will not respond to checkpoint inhibitors. So what I'm showing you here is a large cohort of patients at MGH and Sloan Kettering. This is actually part of Dr. Gaynor's Longevity ALK Positive Award. And here they've looked at 170 ALK positive cancers and looked at the expression of this marker PD-L1. And what you can see is that about 40% of our ALK positive patients have high PD-L1 expression. And in the general field of lung cancer, that means that those patients should be hopefully responsive to checkpoint inhibitors, but that's actually not the case for ALK. And I'm going to show you some data on that and explain why in a moment. But just to sort of settle that question, the reason is that oncogenes like ALK that are signaling constantly will drive up expression of PD-L1 on the cells, but that has no reflection on the cancer cell actively suppressing the immune system. So as I said, for ALK positive patients, the PD-L1 score is not as meaningful as in other patients who have sort of the more typical smoker's lung cancer. Now, what have we seen? So we've seen across numerous studies, trials, retrospective studies, that ALK positive patients do not respond to checkpoint inhibitors. This is 2 large trials that actually are focusing on never smokers, since most of ALK positive patients are never smokers, and showing that for these large trials, even with high PD-L1, never smokers are really not deriving much benefit from these checkpoint inhibitors. This is actually one of the few studies, prospective clinical trials that enrolled ALK-positive patients to checkpoint inhibitors. This one was durvalumab, which is a PD-L1 inhibitor. And what we're showing here are waterfall plots for the group of EGFR and ALK patients who received durvalumab. Some of them had lower PD-L1 expression and some had higher PD-L1 expression. You can see there were some responses as indicated by these downward bars, but when we went back and looked by genotype, there were not a single ALK patient actually had a response to this checkpoint inhibitor. And that's pretty much held up through all of the studies. This is a retrospective study from Mass General that we did. Here again, we've combined EGFR and ALK. None of the ALK positive patients responded to checkpoint inhibitors. And shown here is a recently published study from the led by a European investigator. They call it the Immuno target registry, where they looked retrospectively at a lot of different patients who had these different oncogenes. And they looked at the response rate to checkpoint inhibitors. And you can see for ALK, there are 23 ALK patients in this registry, and again, not a single response to a checkpoint inhibitor. Now, why might this be? Well, a couple reasons. One is many of you know that as a never smoker or a light smoker, your tumors or cancers are just not as complicated as patients who have had a long smoking history. You haven't been exposed to those carcinogens over many years, so there are really many, many fewer mutations within an ALK-positive lung cancer compared to a smoking lung cancer. And so what we call the tumor mutation burden, or TMB, is much, much lower in an ALK-positive lung cancer, and we believe that because there are fewer mutations in these cancers, they just don't look that different or foreign to the immune system, so they're harder for the immune system to recognize. In addition, what we've shown, as illustrated here, is that there's just not that many immune cells around in an ALK-positive lung cancer. For a typical patient, for example, one who has a KRAS mutation, often seen in smokers, we often see lots of CD8-positive tumor-infiltrating lymphocytes. Those are the T cells that can kill the cancer cells. You often can see many of them as shown by these brown dots. But for many of our ALK-positive lung cancers, we see almost no T cells infiltrating the tumor. So between the tumor not looking very foreign to the immune system and also there not being very many immune cells around, that's part of the reason why we believe checkpoint inhibitors are just not very effective for ALK-positive lung cancer. Now you may ask, what about combining immunotherapy with chemotherapy? That's certainly all over the place. We use that front line for patients who don't have EGFR or ALK. We use chemotherapy combined with Keytruda for so many of our patients. And again, just some clinical trial data where we have studied different combinations of chemo with checkpoint inhibitors. You may have heard, many of you have asked me about one of the studies, which is Empower 150, and it's a complicated quadruplet regimen of chemotherapy with this immunotherapy called atezolizumab, which is Tecentriq. And there was a lot of excitement early on because this trial did enroll EGFR and ALK patients, not that many, but they did have some. And the report of this group of EGFR and ALK patients was positive in that it suggested there was a benefit to adding the immunotherapy to chemotherapy. However, when they went back and looked more carefully, it seems that all of that benefit was in the EGFR patients and not in the ALK patients. So again, it doesn't seem like adding immunotherapy to chemotherapy will help in ALK-positive lung cancer. And that's true for this other big study, Empower 130, as well, that again, we didn't actually see benefit to adding immune therapy. So what do we do if we don't have checkpoint inhibitors to use or chemotherapy combined with checkpoint inhibitors? What are our options? Well, we're hoping to find these options. And so this is, in fact, the subject of Dr. Gaynor and Dr. Riley's Longevity ALK Positive Award, where they're really hoping to take a multi-pronged approach to studying the immune system in ALK positive lung cancer specifically. So studying the immune microenvironment, trying to identify, are there any immune cells there at all, or why aren't there more immune cells in ALK positive lung cancers? Also studying the tumor itself to see if there are any mutations there, are they creating any mutant proteins that can serve as potent inducers of an immune response. And then many of our patients will go on to various therapies, including some immunotherapies, so having all of this information correlated with clinical outcome. And the reason I really love this proposal and this work they're doing is that they have a very clear idea, several hypotheses that could explain why so far ALK-positive patients don't respond to checkpoint inhibitors and what we can do to overcome that. So as I mentioned before, we don't see many immune cells. If that really is confirmed in this large study, there are strategies that we can take to try and bring in more immune cells closer to the cancer. If there are other checkpoints besides PD-1, PD-L1 involved that are keeping the immune system at bay, and we identify that in these studies, then there are combinations that we can develop of dual or even 3 checkpoint inhibitors that we could try and use to overcome this primary resistance. And so again, specific ideas about what could really be keeping the immune system from working better in ALK-positive lung cancer, and then specific strategies to try and overcome those. So I would stay tuned because lots of information hopefully will be coming out from this project in the next year or 2. Okay, finally, it's just a few thoughts on future therapies beyond just the immune system. This one's relevant to some patients here in the audience who've had this experience of being on multiple ALK inhibitors, developing resistance, and then developing this complex array of ALK mutations, which I alluded to before. This particular patient had been on crizotinib, brigatinib, and then lorlatinib, had undergone multiple biopsies along the way because he had a palpable lymph node and he wanted to have it resected every time it started to grow. So we had this incredible collection of tumor specimens where we could completely identify the genetic determinants of resistance here. And you can see what happened. He had multiple ALK mutations come out, double ALK mutations, and then ultimately, when he relapsed on lorlatinib, he had this very complicated triple ALK mutation. And so it's very different than what we see when patients fail alectinib, where we only see these single ALK mutations shown here. What we see with lorlatinib, once you've been on sequential ALK inhibitors, including lorlatinib, you now pull out all of these different double and triple ALK mutations. It's really, really complex. And our worry is that right now, for most of these, not all of these, but for most of these, we don't have another ALK inhibitor that can overcome them. And so what I'm hoping to work on again, actually I am working on now and hoping to keep working on for the next year or 2 is to bring new ALK-targeted therapies to the clinic that can try and overcome this complex array of mutations. So this may be in the form of a 4th-generation ALK inhibitor. This may be in the form of a drug that actually binds to a different part of ALK, not the active site we were focusing on, but a different part that can also inhibit ALK, or a broader approach that just focuses on degrading all of the ALK around, independent of the mutations. So I think you should stay tuned for these types of strategies as well, and they're really being developed to address this increasing complexity of ALK mutations that we're seeing in the clinic. The second point I'll mention is this problem of ALK-independent mechanisms of resistance. I told you already, if you don't have an ALK resistance mutation, then oftentimes there's another pathway that's been turned on. And this is a phenomenon we're seeing that in the very beginning, when there's a newly diagnosed patient who's never seen an ALK inhibitor, all of their cancer essentially is very responsive to an ALK inhibitor. So that's what I mean by this big red circle. And then as you become increasingly resistant to these ALK inhibitors, we see that there's a smaller and smaller proportion of patients who still remain sensitive to ALK inhibitors. And the reason we lose these patients here in terms of ALK sensitivity is because they have these other bypass pathways or alternative pathways activated. This is really, really important because even if we do ultimately develop a 4th-generation drug or an allosteric drug, I feel like ultimately the cancers may be able to figure out other pathways to activate. So we need combinations that can overcome ALK, but also overcome these bypass pathways. There have been many that have been reported in the literature by many groups, including actually Christine Lovely and others, and I've listed only some of them here. But they do suggest potential combination strategies. Some of these have entered the clinic already, and we will be bringing this newest set of combinations to the clinic hopefully in the next few months, where we're looking at various combinations of lorlatinib with other drugs, crizotinib, a MEK inhibitor or a SHP2 inhibitor, specifically to overcome this problem of bypass pathways or alternative pathways that drive resistance. Okay, so I'm going to end there because I told you a ton of information, and I want to make sure we have lots of time for Q&A, but I do want to spend a minute on acknowledging all of the people who have done the work that I've shown you today. It's really amazing. I don't know if you guys realize there's like a huge team at Mass General and at other institutions who are all focused on ALK and trying to make discoveries, often in the lab, and to then a whole other team working with them to bring those discoveries to the clinic. So I've shown you some of the people in our group at the MGH Cancer Center who've been working with me on this. I also have to thank all of our funders, acknowledging, we really couldn't do most of this work without funding. So certainly NIH has been funding a lot of our research, but many of the foundations, and I specifically wanted to call out Longevity because they have been so amazing to partner with and to focus on these ALK-related projects. Of course, I have to thank all of you and your families for being part of the research as well. None of this research really could have happened without the patients, right? We couldn't have understood resistance as well as we do now without those biopsy samples. We couldn't have tested any of these drugs without patients volunteering to participate in these trials. So you guys are a huge part of the story and a huge part of the success of these drugs. And then finally, of course, I want to mention the whole ALK Positive community in general for being such an amazing resource. I can't tell you how many patients I see who were lost for a period of time. They didn't understand what was going on with their treatment, or they were frustrated by something going on, and they found the ALK Positive community, and all of a sudden they had resources, they had friends, they had people to ask, and it's amazing. So I wanted to thank all of you, thank the whole ALK Positive community for everything you do for patient awareness, patient education, patient support, and of course supporting all of the investigators as well. Thank you very much. I'm happy to take questions. Please sit.
And we as a group want to make sure that we acknowledge you. Thanks for all of your efforts and all of your time that you give each and every one of us. So thank you very much. That's for you.
Any questions or go straight to
We're going to go straight into the Q&A.